There's a question on please elaborate theranostics approach. So I think this we have mentioned briefly. So what we mean by theranostics is essentially you want to have the capability of both doing a drug delivery as well as tracking. So there's an imaging component also associated with it. Is it a useful approach to tackle the coronavirus? Okay, so that's, I guess, the question right now. So there are a lot of strategies from the biomaterial nanoparticle side, which can have an impact on coronavirus as well as looking at therapies. But what needs to be kept in mind is that given our current pandemic situation, what is the translational potential of any technology? So we want to give preference to those technologies that are quick to translate. Otherwise, that journey itself can be very long. Okay. How can we overcome the protein corona formation around targeted drug delivery systems? So this is particularly in terms of how you design your shell of your material. So the interaction between proteins, which give you the corona, particularly in systemic delivery. So this is again route specific. So if you're doing an intravenous delivery, then that optimization is required with relation to the shell and with relation to the amount of stealth characteristics that you build into your system. I'm trying to see what, okay. How to control the presence of ligand on surface of targeted liposomes. Okay, so this is driven by the chemistry. So essentially, you have choices of which ligand and how much, the density, as well as their orientation and presence, right? So if you have a preformed particle and then cause a linkage to its surface, then you can control the chemistry as to where that ligand is going to be linked. If the ligand is pre-attached, then of course it is going to be prior to self-assembly and is going to be present all over, right? So usual strategy for ligands on targeted systems would be to first create the non-targeted system and then have a specific chemistry to control the targeting moiety on the surface. How liquid crystals are used in drug delivery. Okay, so basically there are many applications for liquid crystals in drug delivery. The main advantage that you get is that of controlling their phases and to have control both on stability and leakiness depending on your required um, entities. Uh, I want to know, is there any critical size of material to cross endothelial cell barrier or any other critical properties to be incorporated? Okay, so as far as crossing endothelial barrier is concerned, I am assuming right now that you mean the enhanced permeation retention effect. It sort of differs depending on what you're dealing with. If you're looking at brain capillaries, it's a completely different story. But for endothelial cells in cancer situations and wanting to exploit the enhanced permeation retention effects, typical ranges of 100 to 500 nanometers have been explored. Again, it depends on your type of tumor, but that's a good first step. What type of materials are useful for forming aerosol carriers? Okay, so for aerosols, you can use, you have to use degradable materials. Usually spherical materials are preferred to, anything which is rod shaped or anisotropic. There appears to be an intense fibrotic reaction to rod-shaped particles in the lungs. And otherwise, you can use polymers, you can use biopolymers, you can use phospholipids, combinations of these. But crucial is you have to optimize them for a couple of things. One, as I mentioned, the mass median aerodynamic diameter of the aerosols that are formed. So there needs to be a certain amount of surface activity to cause respirable aerosols. And two, interactions with the alveolar surfactant, which will determine the response that you will get at the alveolar level. Okay, what is ultrasound contrasting materials? Okay, so these are basically materials which are going to have a gas core. So they are often referred to as bubbles. And what they do is that they enhance the contrast of the ultrasound. And as I explained to you, if you design them suitably, you can also load drug onto them or link them to drug carriers. And by changing the focus and intensity of your ultrasound, you can cause these to burst in certain places and hence cause a release of drug at that site. Additionally, since they also lead to sonoporation, you can then get enhanced uptake of the drug across a barrier. It's very interesting that this has also been used to, transiently open the blood-brain barrier and get actives across to the brain. Okay, so it opens up a whole lot of possibilities, and the advantage is that a lot is known about its safety in terms of ultrasound. A question regarding protein adsorption. Will that have effect on efficacy of drug release? Yes, definitely it has. So when you design in vitro tests or systems, the idea is that you should try to make them mimicking the in vivo environment. The closer you can do that, the more realistic your data will be, and will allow you to quickly go from in vitro to a small animal model. Okay, what will be the fate of a receptor targeted drug delivery vehicle that releases drug at around pH 5, which is usually found in the vicinity of a tumor as well as endosomes of any cell? So will the vehicle release the drug in the vicinity of the tumor, or it will deliver the drug inside the cell? Okay, so this completely depends on, as I mentioned, what is the trigger at which you will cause the release, right? And which part of your carrier is responding to that trigger? So is it that you have just a single matrix system where presence of pH bursts it open? Or do you have core shell systems and layered systems so that some parts can respond to different stimuli? Having said that, usually tumor pHs, we don't look at 5. We look in the range of around 6 to 6.5. So if you want to target release in the vicinity of the tumor, you don't want to actually go all the way down to pH 5, but it's completely dependent on the specific route and application. If you want to look at normal cells and then look at delivery at certain compartments within the cells, you can do so using this concept for endosomal delivery. Okay, could you please emphasize on the preferential I think, route of administration of drug to the respiratory system if carbon dots are being considered? Well, for carbon dots, I would hesitate to give you a specific route because you would have to really optimize in terms of, responses. Since less is known about their effects on aerosols, you may, sort of want to look at systemic routes. Okay. What polymers are used for microneedle preparation, and what about mechanical stability? So there are an entire range of polymers that can be used, and mechanical stability is a big point. You must ensure that they withstand the mechanical forces, not just by themselves, but also are easy to administer. So they should have the mechanical stability to pierce through skin and not bend and break at that point. So that requires substantial optimization. How will you control endosomal escape? So there are various strategies that can be built into the biomaterial carrier for endosomal escape. So some of which are essentially determined by change of phase as soon as the endosome is reached. With the change in pH, there's a change of phase of the biomaterial. And one example being lamellar to inverted hexagonal. So when that happens, you get an inside out conformation, and you come out of the endosome and into the cytoplasm. What are the suitable receptors that can be targeted? It depends on what is the disease. Okay. So you have to look at the disease and look at what are the receptors that are upregulated in that particular disease as compared to the normal. Recently, it is about you doing a nasal gel. Okay. Uh, I have purposely left results out of this scope of this work because I'm trying to, sort of give you concepts. But essentially, it is possible for us to design various kinds of carriers and depots depending on the route of administration and the residence time that is required. And the nasal gel strategy is just one among many that we are actually doing. And the idea being that it should have a localized depot. How to prepare microneedle patches? Okay, this really depends on the type of microneedle patches. So as I mentioned to you, they range from hollow to complete solid to degradable. Some are metallic, some are ceramic, some are polymeric, and it varies. The entire thing varies based on these materials. And there is, of course, a design that has to be done in terms of the, sort of the fabrication of the geometry of your microneedle arrays, which is usually done by, various nanofabrication techniques. How will you prevent from lysosomal enzyme degradation? Similar to the endosomal escape, concept. Here, your concept is that of enzymes. So once again, you sort of, want to either have an impermeant shell which does not react to that enzyme, or an escape strategy which gets away from that microenvironment. Are externally controlled systems preferred over internally controlled? Uh, Unfortunately, in the real world, there's no one answer to something being preferred over another. So it is all specific to which trigger, which application, feasibility, and how that's been modulated. They each have their advantages for sure. And I think that's what you need to keep in mind. So it is possible for you to design some very smart externally triggered systems. But at the same time, internally triggered systems do have some advantages of their own. How to take, I guess this is probably, we may not have time for many more, but how to take drug release imaging in case of external triggered biomaterials. Okay, so essentially this is based on the material properties. So if your carrier or material is ultrasound responsive, then you will get an ultrasound contrast, which is then giving you the imaging. Similarly, if you're using gold particles, you can use near infrared light, as well as you could use CT as a contrast for imaging. And even for magnetic nanoparticles, then it can be used with MRI. Diksha, do we have time for me to do more, or?
You can take 3, 4 more questions.
Okay, okay, all right. Can polylactic acid hydrogel with some useful drug be used to deliver drug efficiently? Sure. Why we don't emphasize more for extracellular matrix triggering mechanism. So I think it's all, very promising. What is required is to get into the details and the specifics. I think there's a lot of potential. The task is to make sure that you design appropriate to the condition that you are trying to actually address. And the closer you mimic the in vivo situation during your optimization stages in your in vitro experiments, the better your chances are of actually getting something which will work even in the in vivo situation. How to evaluate the endosomal escape of the nanocarrier? You basically have to quantify across different compartments your drug. And you can also do image tracking through compartments. Natural polymer or synthetic polymer nanoparticles, which one is better for theranostics? Answer is neither. Basically, the theranostic component is going to be determined by the biomaterial, which is going to respond to an imaging agent. The carrier is going to depend on your route as well as on your application, which disease. So uh, you can have more than one opportunities for this, but the theranostic component is going to be responsive to the imaging modality. Examples of biomaterials. So many have come in the questions themselves, right? Polylactic acid, polymers, phospholipids, some forms of ceramics, composites. Yeah, there are many such examples. Can we use the ultrasound method for cancer therapeutics? And if studies have been undertaken, what are the challenges? Yes, we can use, and in fact, we have done a lot of work in this area. Uh, there are also clinical trials which have shown that if you use ultrasound responsive carriers along with conventional drugs, you can actually enhance the localization, I mean the target concentrations and effectiveness of existing drugs. What are the challenges? Challenges are essentially to, have a very stable form of the bubbles, which can also have sufficient drug loading and can be utilized for this purpose without, building too much on the complexity, because the moment you enhance the complexity, your translational path becomes longer. So I would say that's the major challenge. How can we say that a particular molecule will show photodynamic therapy? You have to actually test it. Yeah. So your trigger is going to be light based, and accordingly, you have to show whether in the presence of your photo trigger, you are getting the trigger responsive effects, and what are the temperatures that are reached by this. and also what is the cell killing or cytotoxicity associated with this phenomenon. Can you share? I think ACS would actually be having this somewhere on the site. Can you share this presentation? So I think it is archived somewhere, right?
Yes, so we are planning to share these presentations very soon on our India landing page. So all the recipients should be getting an email in a week or two time.
Okay.
So slides and your talk and Q&A, everything we are going to uploading.
What type of nanomaterial, biomaterial was used for drug delivery? So every possible type. Okay. So essentially, there's an entire range of materials, and even for the same route, depending on different drugs or different diseases, your choice of material may have to change. And what is important is to try to understand what are the criteria that you're trying to optimize for a particular system, and then design your biomaterial strategy accordingly. Okay, so I'm only taking the questions. Thank you for all the very nice comments and appreciations. I'm not reading those out. Ma'am, do the biomaterials act as inhibitors or disrupt the cell structure? What is the mode of action? So again, it is there's no one mode of action. It is specific to each case. In the case of efflux pumps, which I had mentioned, they are actually acting as either inhibitors or bypassing the efflux pump. As far as disruption of cell structure is concerned, when you give ultrasound or light pulses, you can, at certain intensities, disrupt cell structures if you want to. But usually the mode of action is what I explained with relevance to the envelope structure, which is protection and controlled opening, controlled leakiness, and controlled distribution, giving it specificity as well as a kinetics for release as per the disease site. What should be the approach if we get very sustained drug release at desired site, but comparatively fast release at normal pH when a peptide gel is used as a carrier? So if that is your problem, what is the essential optimization that you have to still do is that you must have minimum leakiness at the non-trigger condition. So if your physiological pH in normal sites is your non-trigger condition, you have to increase the rigidity or packing of your carrier so that this baseline is reduced, because otherwise it is not going to give you specificity of action. You will get toxicity wherever you release the drug in the normal site. What are the advanced studies going on in photothermal therapy? Is it better than other therapeutic techniques? So photothermal therapy, uh, as I mentioned, there's a lot of work going on in terms of having gold nanoparticle based strategies. And the main aspect there is one, to ensure that you get the desired photothermal effects, which means a localized increase in temperature. Two, to avoid toxicity by having large increases in temperature at other sites. And three, to ensure the fate of these gold particles so that finally they should be excreted out, so that you don't get concepts of cumulative toxicity. So that is the optimization that's required. What are the drawbacks of polymeric micelles for drug delivery? You may have limitations in terms of the payload. And depending on your route of administration, you will have to see which polymeric micelle gives you certain types of permeation. you may require some enhancement on those fronts. In case of polymeric core shell particles for dual drug delivery, how to retain efficient delivery to target organ? I'm not sure I understand the question, but essentially, when you have a core shell particle for two drugs, your optimization is for each drug as well as for their combinations and potential interactions. So all of that has to be controlled so that you are going to have usually a controlled degradation of your shell and core, giving you variations in drug delivery. And the efficiency will completely depend on synergism between the two drugs. If 2D or 3D material has one dimension within 100 nanometers, while others are around 1000 nanometers, can they be considered as nanomaterials and used in similar strategy? By as far as definition is concerned, yes, they can be considered as nanomaterials. Uh, in terms of strategy, it has to be specifically looked into, right? So where you're going, which barrier it's passing, and how it will do so. Yes, pharmacoengineering approaches give you good passive as well as active triggering. Yes, that's correct. What are the disadvantages of targeted drug delivery? So I won't say disadvantage, but challenge for sure. If you're using you know a bioligand for targeting, then there are additional sort of complications in terms of translation. One is in terms of how efficient they are finally in the human environment, where different cancers of different patients have variable sort of upregulation of these receptors, and even sort of different subtypes of the same tumor also has variability. So that is one thing that makes a difference. And second, of course, is the challenge of having the efficient stability of the ligand in the final clinical scenario. How do we make drug on a chip system? Do we install tech into a hydrogel? Uh, I am not sure what you mean by drug on a chip. So I will assume that you're trying to have a sort of a model where you want to check drug effects. So typically the hydrogels can be used. You can also use what is called as spheroids, which are 3D aggregations of cells within a hydrogel matrix. And the advantage that you get is that you have barriers of extracellular matrix and permeation, which are closer to in vivo than just a 2D monolayer cell culture system. How do you control porosity of biomaterials for endoserial drug delivery or at any specific? Porosity is usually controlled either by use of porogens, which are going to be of defined sizes, or based on the lyophilization that you are going to utilize, or cross-linking densities. These are common ways in which porosity can be controlled.